Okay, so let's continue on with biochem. So last time we defined the different types of bonds, the difference between ionic, covalent, and hydrogen bonding. Now, because this is biochemistry, all of the groups of molecules that we're going to talk about have carbon in them. So what makes something an organic molecule is that it contains carbon. What makes it bioorganic is that it's a molecule that is found in biological systems, okay, in living things, basically. Or non-living, because we have classes of microorganisms that fit into that category as well. So at the basis of everything is carbon. And if you can remember back to your chemistry class that you took, what makes an atom reactive is how many of the subatomic particles called electrons are circulating in the outermost shell around the nucleus of the atom. So here we have the nucleus of carbon, and carbon has an electron shell that has 4 electrons. And the second electron shell, which carbon has, wants to have 8 electrons. So carbon is basically half full, and that makes it very reactive. It really, really wants to form bonds with other molecules because it's only half full, and what it desires is to have a full electron shell, meaning 8 electrons. Okay, so carbon is kind of interesting. If you take carbon molecules and you put them in flat sheets, you end up with graphite, or what is pencil lead. If you put them in more of a crystalline structure and then put them under a lot of pressure, you end up with diamonds. So you can have molecules that are just carbon, arrange them differently, and you end up with completely different things. So, that makes it kind of interesting. So as I mentioned, organic chemistry means that the molecule has carbon in it. But you can also have things that don't belong in living systems that are organic. So for example, if a person becomes, a scientist that studies petroleum products, like gas or oil, those are organic compounds, but they're not bioorganic. So what we're going to study, and the groups that we're going to go through briefly, are compounds that are found in living systems. And I guarantee you've heard of the majority of them because we see them on our nutrition labels all the time. So we're going to talk about carbs, lipids, proteins, and then very briefly, I'm going to mention nucleic acids. And that's just because we're going to do a whole chapter on DNA and RNA. So, I won't mention them very much here. So when we're talking about biochemistry, the 4 major classes of compounds are what I just mentioned before. You have carbohydrates, which are shown here, lipids, which are shown here, amino acids and proteins, and then nucleic acids. Okay, so one thing you don't have to worry about, I'm never going to take one of these photos and then put them on a quiz or an exam and say, what is this? Okay, all I want you to see from this diagram is that these are big, complex molecules, and probably lipids are the biggest and most complex. But we're not going to worry about structure and how they bond, in great detail, simply because this isn't a class in biochem. We're just going to learn about these compounds because I'll talk about them more because they're involved in how microorganisms reproduce, how they utilize energy from us. And so that's what I want you to be familiar with. So one of the things I want to talk about is how they're organized, okay? And there's a couple of terms I want you to understand. So if you ever see the term monomer, mono means one, okay? So when I talk about a monomer of a biomolecule, such as carbohydrates, lipids, etc., what I'm talking about is that it's the subunit that makes up the larger molecule, okay? So monomer means the same thing as subunit. We also call it building block. I might ask you a question, what is the building block of carbohydrates? And so that's asking you what the monomer is or what the subunit is. So when you take many monomers and hook them together, that's called a polymer. Poly means many, okay? So taking monomers of carbohydrates, hooking them all together, you end up with a polymer, which is a carbohydrate. So that's where the large class comes from. Okay, also the differentiation between digestion and synthesis. Okay, so digestion is when you go from a polymer and cut it up into its monomers. Okay, so I want to make sure this is clear. And actually, let me add something in here. So let me see if I can do this, blah, blah, blah. If it'll let me. Oh, I don't think it will. Okay, so I'm going to add this to the PowerPoint just so it's clear. But digestion means taking a polymer, digesting it, and going and producing monomers. So when I say going from many to one, that means taking one polymer and digesting it so there are actually many polymers. So it's a little confusing, so I'm going to make sure that I change this so it's more clear. Synthesis is when you take monomers and put them together to produce polymers. So, for example, if you think of eating a cheeseburger, okay, so a cheeseburger, we're going to pretend that it's made mostly of protein. So it goes into your stomach. Your stomach acid helps digest it. So you're going from a large protein into many amino acids, which then get absorbed in your small intestine, travel through your bloodstream to your cells, and then your cells can take all of those individual amino acids and put them together in whatever protein they need to help their function. So that's the difference between digestion and synthesis. So if we look at the four classes, I want you to know the larger classes, so the polymer, and know what their monomers are. Okay, so carbohydrates are also known as polysaccharides. Saccharide is just the fancy word for sugar. Okay, so a carbohydrate is a polysaccharide in that it's, many sugar molecules hooked together. And the building block or the monomer are referred to as sugars. A lot of times we call them simple sugars. For lipids are probably have the most number of names. We can call them fats, lipids, membranes. I usually refer to them as fats or lipids. And for simplicity's sake, we're going to say that the building block or the monomer are fatty acids. Okay, proteins are pretty easy. Protein is the polymer. They are made up of building blocks called amino acids. And then nucleic acids, such as DNA or RNA, are made up of building blocks called nucleotides. Okay, so that's the difference. But yes, you should know what the polymer is and know what the building block or monomers are of each of these four classes. Okay, so let's start with these. So the first big class are carbohydrates. And they're my favorite because guess what? They're delicious, okay? It's the reason, that I drink a lot of sugary drinks, that and caffeine. Carbohydrates are basically sugar, and their primary function is to provide fast energy. So in terms of what they're made up, in terms of atoms, they're actually really pretty simple. They have carbon, they have hydrogen, and they have oxygen. And a lot of the building blocks have this little kind of spaceship structure, and then they can be hooked together to form carbohydrates. So, for example, if you're looking to measure, blood sugar in a patient, what you're looking to measure is the amount of glucose. Glucose is a simple sugar or a building block of carbohydrates. Fructose, you hear about, in terms of diet, high fructose corn syrup, which can be found in a lot of products. Galactose is also a very common simple sugar. I want to point these out because DNA and RNA have a sugar component. And so DNA has deoxyribose. That's where the D comes from in DNA. And RNA, which starts with an R, has ribose. That's the difference between the two. But yeah, if you hook them together, then you end up with carbohydrates or polysaccharides. So one of the ways that animal cells store sugar, which can then be broken down and burned to provide energy, is in the form of glycogen. So what glycogen is it is lots and lots of glucose monomers hooked together, and they're hooked together in chains, and then they're branching out here, and that's what's stored in our liver. So, for example, if you're a person who likes to run marathons or long races, which, God, I don't know why a person would want to do that, but hey, God love you. A lot of times runners before a race, you'll see them eating a lot of pasta, and they're basically doing what is called carb loading. And it's so their liver becomes packed with glycogen. And so therefore, they're going to have lots of glucose molecules to be able to burn during the race when they're not able to eat, okay, to provide them fast energy. So carbohydrates are great for that. The only problem is our liver has a limited capacity. It can only store so much glycogen. And then in a cruel prank from nature, any extra sugar that you take in, once your liver is full, it gets turned into fat, which is much, much harder to break down. And that's why people who eat tons of excess sugar gain weight, and it's hard to get rid of. So, unfortunately, that's what nature decided. But yeah, we store it for fast energy. Okay, so I'm at 11 minutes. I'm going to stop and get this uploading, and then I'm going to pick up with lipids, our next category. Thanks.